All right, so let's get started. Good morning, everyone, and thanks for joining us today. We're glad you could join us for our virtual research seminar series. My name is Camilo Ansara. I'm Associate Director of Business Development and Industry Relations at the MASH Academy, Keck School of Medicine. And for those of you who are not yet familiar with the MASH Academy, our mission is to empower the convergence of research disciplines to address challenges in human health and disease. We're here as a dedicated team to help facilitate connections and collaborations across USC with the School of Medicine, with other researchers, as well as external industry. And we are very excited to be able to bring this seminar to you today. But before we get started, just wanted to mention a few housekeeping items. So following this presentation, we'll have a brief Q&A session. And for the Q&A session, I would like to ask that you please use the raise your hand function. When you do so, I will be able to call on you and allow you to speak, and you'll be able to address the speaker directly and ask your question. If you prefer, you may also use the Q&A tool to write in a question, but we encourage you, if possible, to raise your hand just because you will be able to ask the question directly to the speaker and the context of the question is better understood. And finally, I also wanted to let you know that this webinar is being recorded. So both the slides and the recording of the presentation will be available on the MASH website within a few days. So it is now my honor to introduce our speaker, Professor Mo O'Gorman, Chief of Laboratory Medicine at CHLA and Professor of Pathology and Pediatrics at the Keck School of Medicine at USC. Professor O'Gorman's presentation today is entitled Lymphocyte Subset Counts in COVID-19 Patients, a Meta-Analysis. Okay, so let's get things started. We have a really great presentation today, and it is my honor to introduce our speaker. So please welcome Professor O'Gorman.
Good morning. I trust that you can all hear me. First of all, I want to thank the MASH Academy for the invitation to speak this morning. This is the second presentation coming out of CHLA. As the Chief of Laboratory Medicine, this pandemic has hit our department particularly hard. We were up early with viral sequencing, serology, and in fact, whole genome sequencing, which we continue to do and remain very busy. We have not had the directive to work from home because we couldn't. And so what I thought I would present today is a work that we have recently completed in terms of evaluating lymphocyte subset publications. As an immunologist, it's fairly compelling to me that we try to understand the immunopathogenesis of this disease, of which we still know very little. And so what I thought I would do, given that we had presented earlier, and to just kind of give an indication of how fast and rapidly this pandemic is moving, I thought I would give an update on the pandemic, and with a particular eye of looking at the case fatality rates. And what I find particularly interesting is the differences in case fatality rates locally, nationally, and internationally across different countries. Then very briefly, and I'll apologize to anyone who is a molecular virologist or studies viral pathogenesis, because it's going to be a very shallow look at basically viral replication and immunopathogenesis, and how are we getting to the point where some folks seem to manage the virus even without symptoms, while others go on to a very dire pneumonia and death. If we're going to try to differentiate those patients that are going to survive the infection versus those that succumb, we're going to have to try to find biomarkers to help us identify those patients that would go on to more intense treatment versus those that may recover on their own. And so as part of this, I'm going to look at some of the biomarkers, mostly delving into the study that we're going to talk about today that we recently published a few weeks ago. And that's a meta-analysis looking at lymphocyte subset measurements in patients infected with SARS-CoV-2 and suffering from COVID-19 disease. So the last time we presented was April 27th. It was a challenging time where the US had taken over from China, and Europe was suffering massive increase in the daily number of reported confirmed cases. So as you can see, on April 27th, there were only what we can say now is only 3 million, and at the time we were thinking was catastrophic. US out in front with Europe and the Middle East starting to pick up in numbers, to just this week where the cases worldwide are now, as of today, approaching 14 million, with the US still leading, Brazil catching up, and now we're seeing it in Asia, Russia, and Peru as the next top caseloads, where the pandemic is still fulminant.
Sorry, Dr. Oberman, can you share your screen, please?
I am sorry that you are not seeing my screen. I will go back. I apologize. Okay. It tells me that I'm sharing. Let me try this one more time. Okay, I'm gonna have to get out. Can you confirm that it is now sharing?
It is not showing your slides. Yeah, there you go. You're in the
okay.
Slide, yeah.
I apologize. It was telling me that it was sharing, and now I can see it's a challenge with split screens with several. All right, so we'll pick up. This is the introduction. As I mentioned, we were at the update of the pandemic, and we were just going to get into case fatality rates. We're then going to briefly discuss COVID-19 immunopathogenesis, and then biomarkers to help us try to predict those patients that will go on to severe disease or die, or those that may respond to treatment, or markers of response to treatment, viral clearance, or in fact recovery. And then we're going to delve into the subject matter of this lecture is looking at lymphocyte subsets in patients infected with SARS-CoV-2 and suffering from COVID-19 disease. And again, I apologize for those of you who did not see this. But as I mentioned, the last lecture that I gave to this group was late April, where the case rates were soaring in the US and Europe. We now know that the disease is still raging in the United States, but has now spread and is rapidly increasing in South America and Asia, including Russia, and where the cases are increasing dramatically every day. Associated with this are obviously the case fatality rates, and again, what is fascinating to me is the variability in the case fatality rates. So as you follow the green line on the graph, we're looking at the mortality rate, which is the death rate, the number of deaths divided by the confirmed SARS-CoV-2 cases. And so as you can see that the case fatality rate from Russia being less than 1% to being over 10% in many countries in Europe. And again, we have no explanation for the variability of the case fatality rate, whether it's the virus, whether it's the individual, whether it's the demographics of the patient population, or all of the above are still under intense investigation. So globally, there's an extreme variability in the mortality rate. We are also experiencing the same variability within the United States, where you can see, we follow the green line is the mortality rate due to SARS-CoV-2 and COVID-19 in the United States, where we have in mortality rates that vary from almost 10% to less than 1%. So this was again back in April, and if we look this week, we still see the same extreme variability. The United Kingdom, Mexico leading the way at 15%, again down to the Middle East, where the reported case fatality rates are sometimes below 1%. Within the United States, we are seeing the same thing as you see the massive number of confirmed cases in New York, with California fast approaching the confirmed cases in New York, and a four to five-fold increase between the case fatality rate in Florida and California versus New York and New Jersey. So again, there is currently no explanation for the dramatic differences in the case fatality rates. And if we're going to understand how we're going to get a handle on treating patients infected with SARS-CoV-2, we're going to have to understand the pathogenesis of mortality due to the virus. I thought it would be interesting to compare also where we were in April when we presented last time to where we are today. And to do this, I thought it would be interesting to compare new cases per day over the pandemic in New York versus California. California is in trouble, as you can see. We were all worried about the high numbers per day of new cases that were being reported in New York in April. They went into strict lockdown, and as you can see, have been able to somehow manage the transmission of the virus in their population to the point where they are now literally under 1,000 cases per day. We were watching that, thinking that we were just going to cruise into the cessation of the transmission of the virus. However, with the May-long weekend and the massive protests, which may or may not be attributing to the rapid rise that we've seen, we are seeing a dramatic increase up to and over 10,000 cases per day in California. And this is showing no sign of abating. This can be paralleled when we look at mortality and deaths per day in New York. And we saw the dramatic pictures of the bodies piling up in New York as they reach 700-800 cases per day. Our death rate in California was behind, as you can see, started to pick up. We thought we were getting a handle on it as we plateaued and started to decrease. But again, after the long weekend and the protests, we're seeing an increase. I always find it fascinating that we don't count deaths on weekends, which accounts for the raggedness of our curve. And so again, we are seeing an increase in death. We are seeing a variability in the death rate. And we really do not understand the underlying cause of the differences in the symptoms that patients experience. So the unanswered questions that we're still under investigation today is what causes certain patients to experience severe disease while others remain mildly symptomatic or in fact asymptomatic. One of the things that we are observing here is that the vast majority of our children seem to get infected without any symptoms whatsoever, or in fact very mild symptoms, other than a small cohort of patients that are, the numbers are increasing. In fact, we have approximately 20 patients now who are experiencing what is being referred to as CMIS, or a Kawasaki-like inflammatory disease. So we are seeing that in some of our children. And again, we don't know which children are experiencing these symptoms or why children seem to be protected from severe disease. My bias as an immunologist is that it is the host factors that play a significant role in the pathogenesis of the disease. The cytokine storm is clearly responsible for killing approximately 1/3 of the patients that die with COVID-19. We don't know why they're experiencing a cytokine storm, whereas the person next to them and other family members do not. And so the question that we're asking is, are there, as a laboratorian, are there immunological biomarkers that we can measure that would be predictive of severe disease, poor outcome, recovery from disease, viral clearance, or in fact, just letting us know that the patients are on the road to recovery? So let's just take a quick look. And again, I apologize to those of you who study viral molecular mechanisms of pathogenesis, because this is a very, very shallow look at it. But again, I just want to get to the point where we are going to try to clear the virus and what determines whether that's a normal response or in fact an abnormal response. And again, I'm going to tell you right up front, we don't know what triggers the difference between a massive inflammatory response versus a controlled immune response that eliminates virally infected target cells. So obviously the first step is of the infection and the airborne viral particles binding through the spike protein to the target receptor, which we all know now today, which is very similar to SARS-CoV-1, in fact, is the angiotensin converting enzyme 2. We also know now that it binds through CD147, which is also present on type 2 pneumocytes, although this is less well publicized and not 100% agreeable to all. Following adherence to the angiotensin converting enzyme, or ACE2, the spike protein is rapidly cleaved with the cellular transmembrane serine protease 2. This cleavage is absolutely essential for the spike protein to change its shape into trimeric structure, then allows fusion between the actual viral membrane and cell membranes. Once the virus fuses with the membranes, in fact, the cytoplasm of the virus is open to the contents of the virus. So the new viral nucleocapsid and the small proteins inside the virus are dumped into the cytosol of the infected cell. The viral RNA is rapidly uncoated and quickly undergoes replication and the translation of the polyproteins to generate replication transcription complex. The replication transcription complex then generates two types of RNA, negative stranded RNA, one for the full length genomic RNA of the actual virus that will be packaged into the virus, and then subgenomic messenger RNAs, which are translated into the structural and accessory proteins, which are also packaged into the virus for, again, generating new RNA, structural proteins, et cetera, when they reinfect new cells. So once the genomic RNA, the subgenomic RNA, and all the accessory proteins are translated and replicated, the virus is rapidly assembled and is released by cells via a process we believe of exocytosis. Following the release of the viral particles from essentially lung cells, type 2 pneumocytes, the cells die. Another area of active investigation is the mechanism of cell death. For those of you who are into studying cell death, we now have over 15 individually coded mechanisms of dying. And so from pyroptosis, apoptosis, necrosis, autophagy, netosis, et cetera, the mechanism of the cytopathic viruses are not well understood, particularly with SARS-CoV-2, although there is some consensus that it is a process of pyroptosis, which leads to, or can lead to, a fulminant inflammatory response. So following infection and release of the virus from cells, the innate immune response is activated, which leads to the recruitment of monocyte, macrophage, continued release of cytokines and chemokines, leading to priming and adaptive of our T and B cell responses for the production of a humoral immune response or anti-SARS-CoV-2 antibodies, as well as cytotoxic T cells, which in most cases will result in the clearance of virally infected cells, inhibition of virus by binding, antibody binding to the virus, and clearance by antibody-dependent cellular cytotoxicity, et cetera, ultimately in resolution of the infection. However, and this is what the subject of this lecture is, in some cases a dysfunctional immune response occurs, which leads to a hyperinflammatory state, acute respiratory distress syndrome, or ARDS, which we're hearing so much about, and then respiratory failure and ultimately death. And to date, there seems to be some consensus. This results in approximately 70% of the COVID-related deaths. The other mechanism that we hear about, which is primarily an inflammatory response, or cytokine storm, as it's being referred to, somehow the viral response leads to an abnormal cytokine response, ultimately leading to organ failure, organ damage, and this is about a third of the cases that leads to result in significant morbidity and mortality related to the cytokine storm, up to and including severe coagulopathies that leads to loss of limbs, et cetera. So today, the mechanism by which SARS-CoV-2 subverts the body's innate antiviral responses are not completely understood. And so I copied without permission beautiful slides that were published right around the time that I gave my last lecture, kind of giving you a visual representation of what's happening at the level of the lung mucosa. So as you can see on the left at the top, the virus is going to bind via the spike protein. The spike protein is cleared by TMPRSS2, allowing it to bind to the membrane, and it is brought into the cell by process of endocytosis, where the virus replicates. generates full-length genomic RNA and the polyproteins, which are packaged back into the virus, and it is then shed from the cell, upon which the cell essentially dies. In a normal immune response, this would stimulate the alveolar macrophage to secrete cytokines, which is, leading to an innate response, recruitment of macrophage monocytes and T cells, further stimulating macrophage monocytes and T cells, ultimately secreting interferon gamma as well as other cytokines, which essentially leads to a feedback loop. And at this point, the pathways diverge. And so what you would expect in most cases in a normal individual with a normal response to a cytopathic virus is that the alveolar macrophage would essentially pick up and clear the viral particles via antibody-dependent clearance. The cytotoxic T cells would be primed by the CD4 T helper cells to eliminate virally infected pneumocytes. CD4 cells obviously are helping in the cytotoxic T cell response as well as in the production of antibodies. This would lead to the rapid clearance of the infected cells with a very limited inflammatory response. And this we suspect is what's going on in our asymptomatic or moderately symptomatic patients. And this would be considered a normal effective immune response for the clearance of virally infected cells. What we don't know is what leads to a dysregulated or massive inflammatory response, where the increase in cytokines leads to vascular leakage, an overwhelming cytokine response, and an overwhelming inflammatory response, which leads to excessive infiltration of essentially monocytes, macrophage, and T cells, a systemic cytokine storm, and then pulmonary edema, pneumonia, widespread inflammation, organ damage, and in some patients succumbing to the virus and a massive inflammatory response, either with pulmonary pneumonia or massive organ failure. So how are we going to unravel the difference between a normal protective immune response versus an innate immune response that's out of control? To date, we obviously have to further unravel the actual innate immune response and what that relationship is to a later adaptive immune response. We're going to talk about that later because it does appear that the patients that do succumb have at least abnormal components of your adaptive immune response, particularly a massive decrease in the number of T cells circulating in the peripheral blood. Again, we don't know at which point the normal protective effects of cytokines and their normal role in elaborating an effective immune response become out of control, dysregulated, and eventually lead to overwhelming inflammatory response. We are familiar with a cytokine storm in our patients being treated with new immunotherapy for cancer, where you have a large increase in cytokines, occasionally leading to a cytokine storm and definitely requiring immunosuppression in order to get that under control, or the patients will succumb to the cytokine storm, which can be fatal. We need to know more about the humoral immune response kinetics and protective immunity versus potentially immune enhancing or antibody enhancing effects of antibody. We are starting to see reports of the longevity of the immune response, and there are reports supporting protective immunity, other reports suggesting that the immunity may be short-lasting, at least in terms of antibodies measurable circulating in the peripheral blood. We don't quite understand the role of T cell mediated immunity. Is it our T cells that are in fact leading to the pathogenesis of the virus, or are they protective? And if it's one versus the other, what is the difference? How do we measure the difference? Again, what's the role of exposure to common coronaviruses? One of the theories of why our elderly are succumbing is that they have a massively accumulated immune response to coronavirus. And in fact, as we age, maybe our ability to control those responses have waned somewhat, in which case we're really having an anamnestic response to coronaviruses. There are some similarities between the 4 common coronaviruses that cause relatively mild colds versus this more severe SARS-CoV-2. And again, what is the relevance of T-cell activation versus exhaustion? And as I mentioned earlier, what role do Tregs play in trying to control an out-of-control immune response? And is there a specific effect on Tregs? Are Tregs less functional in adults? Are they less, in elderly? Are they less functional in individuals with ultimately inflammatory disease, heart disease, stroke, diabetes, and obesity? And so I've been interested in studying the literature and in fact proposing studies to look for biomarkers, primarily immunological biomarkers of severe disease and death by comparing those patients that recover or have mild disease or have recovered from the disease to those patients who are admitted with severe disease who are requiring oxygen support for survival. So we now know that there are several blood as well as immunological parameters that look to be predictive in patients who go on to have higher risk for severe complications. These analytes include the acute phase proteins, examples CRP, serum amyloid, procalcitonin and lactate dehydrogenase, as well as D-dimer and ferritin. There's some consistency showing that patients with severe disease have higher levels of these acute phase reactants and markers of inflammation than those patients that have a more moderate or mild disease. Immunological biomarkers are important and have been consistently shown to be increased in those patients who have severe disease versus mild or moderate disease. And several markers have been measured, and there is some consistency showing that the macrophage elaborated cytokine ICP-10, IL-6, IL-8, IL-10, as well as IL-2 receptor, IL-1 and IL-18 are increased. There have been several studies now in the brief meta-analysis that I reviewed just on markers of disease, both blood markers and immunology markers, now approaching 100,000 patients, showing some consistency in basically inflammatory markers as well as immunological markers. So I think CRP and lactate dehydrogenase are leading the way as far as serum markers. D-dimer is not far behind. And then decreased blood platelets and decreased peripheral blood lymphocyte counts combined with normal or elevated neutrophil counts have been particularly well studied, to the point that what we're going to talk about today is lymphopenia in COVID-19. So I'm going to show you in a minute a study that was just published yesterday out of Philadelphia, University of Pennsylvania, showing that lymphopenia is the most common and most frequently observed prognostic marker that appears to predict both mortality and morbidity, even when measured at earlier stages of the disease. And so lymphopenia, it can be measured relatively easily with a common automated lymphocyte leukocyte differential, where you get absolute lymphocyte counts as well as lymphocyte percentages of your white blood cells. It's a relatively simple test and an inexpensive test, and it is extremely valuable in predicting morbidity and mortality. However, if you want to understand lymphocytes, you're going to have to get into immunology, and we refer to the understanding of lymphocyte subsets as immunophenotyping. There are several hundred, maybe thousands of lymphocyte subsets that are now understood, studied, and known, and they are all essentially identified by the relative expression of combinations of different surface markers. So what I'm showing you in this very brief summary of flow cytometry is the most common method of immunophenotyping or measuring lymphocyte subsets. So basically, this is a peripheral blood sample. We now use whole blood so that we're not manipulating any of the lymphocyte subpopulations in any way. You stain whole blood with a combination of monoclonal antibodies, each specific to an individual surface marker. We can now measure up to 15, 20 in a single tube. So we're able to go down to very, very, very specific lymphocyte subsets. And so these markers are tagged with fluorochromes. They are then stained with the individual monoclonal antibodies, all in a single tube, identifying their specific cell surface receptors or markers. You then lyse the red cells, and individually these cells are passed through a flow cytometer where they are queried on a cell-by-cell basis, and the expression of each of the 15 to 20 individual parameters are measured by their capture through finely tuned optical filters, dichroic mirrors, and finally with the light measured in photomultiplier tubes, with the signals being translated from an analog to a digital signal and being expressed in terms of the level of fluorescence in each of the individual parameters. So what we're showing here, following the essentially analysis of a single cell suspension, one by one, thousands of cells per second. This is all graphed in a 2x2 dot plot where the level of fluorescence of each individual parameter is measured on a log scale. So in this diagram, we are actually looking at a 3-parameter differential of white blood cells. So we have lymphocytes who are identified based on the level of expression of leukocyte common antigen, or CD45, in a violet color. Monocytes and granulocytes are differentiated both by the level of CD45 on their cell surface, but also on their innate light scatter properties. As the cells pass through the laser and are interrogated, they scatter light both in a forward and right angle direction. Cells that have interior complexity, lymphocytes look like small beads. Monocytes have a lunar-shaped nucleus as well as some internal granulocytes and internal granules, whereas polymorphonuclear leukocytes or granulocytes have an extremely complicated nucleus and several granules and scatters a lot of light in a right angle direction, so we get a 3-part differential. By flow cytometry, we can then gate on individual populations and look for the expression of specific cell surface markers. So in this case, we're looking at a routine immunophenotyping test, which is measuring total B cells, total T cells, natural killer cells, T helper cells, and T suppressor cells, all identified based on the surface expression of either CD3, CD4, CD8, CD19, or CD16 and 56. So in the first diagram, we're looking at CD3 versus CD1656. All T cells will express CD3. Natural killer cells will not express CD3, but they express bright levels of CD16 and 56. Neither B cells will express neither 1656 or CD3. So our third population will be B cells. We can then gate further on the T cells and separate them into CD4 and CD8 T cells, or T helper cells and T suppressor cells. We can then further gate on lymphocytes and look at CD3 versus CD19 to get T cells and B cells. So this is essentially what we're going to review now in a meta-analysis that I was able to perform with a group of scientists from Becton Dickinson in Northern California. And so when we started looking into this early on as an immunologist and flow cytometrist, I was fascinated to see early reports coming out of China of aberrations in lymphocyte subsets in COVID-19. Further review of the literature actually indicated that this had been looked at in SARS-CoV-1 as well as MERS, with a consistent observation that lymphocyte subsets were affected in patients with severe disease. So I did a literature search in PubMed and found in April 12, 2 weeks later, 31, a week later, 98. So this is exploding. We decided in May that we would submit a manuscript looking at a summary of all the reports. This was submitted in actually May 15th. It was sent back for review, was accepted, submitted in May, and it was accepted 2 weeks later. When I looked again this week, we've again doubled the number of reports looking at COVID-19 and lymphocyte subsets. So this is just a summary picture of our manuscript that was accepted second week of June. It's still not out formally. It's still in the e-format. But basically what we're going to review now is the information investigating lymphocyte subset abnormalities in patients with COVID-19. So basically, as I mentioned, we did a PubMed search for CD19 lymphocyte subsets, which brought up 258 publications. We reviewed each of these publications for 2 criteria that had to be in, or we did not select the publication. In the first instance, it had to give some indication of the clinical characterization of the patient populations being investigated. In the second case, it had to present lymphocyte subset data in terms of the routine immunophenotyping that I presented earlier, T cells, total T cells, T helper cells, cytotoxic T cells, B cells, and natural killer cells. So in all, out of the 258 manuscripts, 16 had both of those criteria in detail that we thought was sufficient enough to review in detail. Interestingly, a Google search after that was done brought up an additional 40 or 50 manuscripts, 4 of which satisfied our criteria of having both the clinical criteria as well as significant lymphocyte subset data. One of the things that we observed was that the way that the disease severity was reported in each of the manuscripts. And so what we decided to do was we separated into 2 categories, either mild and moderate disease or severe and critical disease. The disease status assignments were essentially grouped, and the various names for mild and moderate included mild, survival, moderate, non-aggravated, and non-critical. The severe critical included deceased, non-survival, critical, and aggravated disease. So out of these 20 publications, a total of 3,000 subjects were included. 76% of those were classified as mild, 24% were classified as severe or critical. The sample size of the subjects that had CD4 and CD8 T cell counts ranged from 17 to 500. And then a smaller subset of those actually included the lymphocyte subset data for both B cells and NK cells. What we observed is that the mean cell counts were consistently decreased in our severe critical group, and the differences in the weighted mean value of cells between these 2 groups were significantly different, in fact, for each of the lymphocyte subsets. So we measured fold changes, and as a standardized fold changes between the mild, moderate, and severe groups for both CD4 and CD8, which we noticed were significantly larger. So the differences between the mild and moderate for CD4 and CD8 were dramatically different than the differences for B cell and NK cells. And I'm going to go ahead and show you a graphic representation of that we assembled with the help of our statisticians. So this is a figure summarizing mean cell counts across articles. The actual size of the dot is related to the group of patients in each of the individual publications. And so graphically, you can see in green are the moderate and mild patient group, in red are the severe patients. And you can see with CD4, CD8, B cells, NK cells, and in fact, total lymphocyte counts, a dramatic decrease in the level of absolute lymphocyte subset counts in patients with moderate versus severe disease. This is summarized in the table below with the 95% confidence intervals. You can see that this is significant and consistent in all of the reports that we saw. So the meta-analysis was formed to calculate the standard mean differences in the 95% confidence intervals between the mild, moderate, and severe critical groups for each of these subsets. And the lymphocyte subset standard mean difference was significantly lower, again, as we mentioned, using a standardized mean difference as the calculation. And this is represented here with the mean difference in the 5th and 95th% confidence intervals, with the range of the interval being related to smaller sizes and increased variability within the different studies. So as you can see, in every single study, there is a difference where the absolute counts for CD4 and CD8 are significantly lower in severe versus mild or moderate disease. This held true, but less intensely so for B cells, natural killer cells, and finally for total lymphocytes on the whole. This is a summary of a few of the articles that I found particularly compelling early on in the study. This was the first significant study that presented detailed report of the normal reference ranges in healthy individuals versus the actual absolute counts for total lymphocytes, CD4 T cells, CD8 T cells, B cells, and NK cells. And again, in this study, T cells were significantly lower in the severe versus the moderate patients. And you can see even in the moderate group, the level of absolute counts in patients with moderate disease was significantly below the normal range for T cells, CD4 helper cells, and CD8 cytotoxic cells. The next study I found particularly compelling because this was a prospective study. Each of the studies, other than this study, were retrospective observational studies. This was a prospective study measuring lymphocyte subsets as well as other variables in a multivariate analysis showing the results of several parameters, looking at whether or not individually measured parameters upon admission would be predictive of mortality. As you can see from the table above, CD3, CD8, as well as cardiac troponin were the most predictive biomarkers of death in this cohort of 179 patients measured upon admission. So this is particularly compelling in the sense that this could be something measured early in the disease course. And again, these are just 3 studies quickly summarizing more potential biomarkers for looking early in the disease, potentially predicting elimination of the virus, response to therapy, or in fact, whether or not patients might be on the road to recovery. And again, you can see the sizes in these studies are not large, but they are compelling in the sense that the lymphocyte subset data, in fact, predicted duration of viral clearance, absolute T cell counts, predicted response to therapy early in the treatment course. And finally, it looked as though when patients recovered, their absolute T cell counts, as well as B cell counts, that those patients that recovered their counts, in fact, went on to recover from the disease and survived. So finally, as an overview of the studies and some of the, I think, limitations, the studies that I've reported have all come out of China, which is interesting in its own right. However, we know that this is a global pandemic, and I think that's a limitation of the meta-analysis. There is a developing consensus, and I'm going to show you more about this in a minute, that the decreased absolute lymphocyte count is in fact predictive of disease severity, even in the face of a normal or in fact elevated neutrophil count. So this in and of itself is interesting. And there are new parameters that are combining either lymphocyte subsets or neutrophil counts together. So you have a neutrophil to lymphocyte ratio or a neutrophil to CD8 T cell ratio, for example. They're in fact showing some very strong predictive potential. Absolute T cell counts seem to be preferentially affected in severe versus moderate disease. Increases in absolute T cell counts during hospitalization may be predictive of response to treatment and viral clearance. But again, more studies are required, and more studies are required domestically. This has been so compelling that in fact China has adopted guidelines that recommending that all patients that are admitted for SARS-CoV-2 induced pneumonia have lymphocyte immunophenotyping performed. This is the guidance that has come out in a brief summary, indicating that high attention should be paid on a situation where the absolute value of lymphocytes is less than what we would say 800 per microliter, or 8 times 10 to the 9th per liter, 0.8 times 10 to the 9th per liter. And the number of CD4 and CD8 T cells are significantly decreased. And then they also recommend that this gets measured every 3 days. Uh-oh, I'm losing my, I've lost my, let me see if it recovers. I've lost my PowerPoint. Let's give it 1 second. We're only 3 or 4 slides from the end.
Yeah, let us know. We can we can broadcast ourselves if you have any problems.
Okay. let me close it and restart it and see if that works. Oh, that's interesting. Okay, let me, are you seeing my screen?
We're seeing your screen, but not your presentation.
It's gonna come back up here.
Okay.
I apologize, not sure quite what happened there. All right, are we back?
Yes, we are.
Excellent, I apologize, not sure what happened there. Okay, so just as a summary of the overall studies and our meta-analysis, severity of SARS-CoV-2 disease correlates with low blood albumin, hypertension, thrombocytopenia, increased blood IL-6, and as well as procalcitonin, and we now know lactase dehydrogenase. Lymphopenia appears to correlate with mortality, acute respiratory distress syndrome, and ICU admission. This meta-analysis showed clearly that the level of decreases in CD4 and CD8 T cell predicts moderate versus severe disease. T cell counts may also predict outcomes in terms of ICU admissions, treatment efficacy, viral clearance, and recovery. CD4 and CD8 T cell counts, even in light of all of the biomarkers that are being investigated, seem to be an independent predictor of COVID-19 outcome. And again, a limitation of our analysis is that these were all studies performed in China, and we now know this is a global pandemic. So what can we do? What further studies need to be done? Clearly, we need to see if patients admitted with COVID-19, if measuring some of these parameters can in fact predict the risk of progression to severe disease and or admission to the ICU. If the patient in fact has severe disease, is it in the ICU? Do we have markers that will predict deterioration and mortality? If the patients are being treated, do we have markers that will predict improving immune status, viral clearance, and convalescence? Clearly, the pathogenesis is still under investigation, and the precise mechanisms leading to reduced peripheral blood lymphocyte remains to be fully elucidated. And although we are observing this consistently in our patients, we are still at at a loss for understanding the exact mechanism that would lead to a reduction, severe reduction in peripheral blood lymphocyte counts. And then beyond the major lymphocyte substance, how are other immune cell subsets involved? In fact, there are new studies coming out measuring, as I mentioned earlier, besides the major lymphocyte subsets, there are well-known functional T cell subsets that can be measured. Example, naive and memory B and T cells, regulatory T cells. What about T cell activation markers, markers of senescence and exhaustion? And in fact, can we combine these markers to improve prognostication? And again, these studies remain and are under, some of them are being developed. And in fact, we have two studies that are currently under IRB review in collaboration with our folks at Keck Hospital. This was fascinating because it just came in this morning and I thought I would share it. It really bodes well to support what we have just discussed. Comprehensive mapping of immune perturbations associated with severe COVID-19. And I'm just going to read this last line in the abstract. We found the neutrophil to lymphocyte ratio to be a prognostic biomarker of severe disease and organ failure. Our findings demonstrate broad innate and adaptive leukocyte perturbations that distinguish dysregulated host responses in severe SARS-CoV-2 infection and warrant, again, therapeutic investigation. And then I'm going to end with the last line. Altogether, these data reveal multiple immunophenotypic abnormalities in severe COVID-19 that are not found in donors with moderate or recovered disease and confirm the prognostic utility of neutrophil to lymphocyte ratio or neutrophil to T cell ratio. And so with that, I will thank you for your attention.
Thank you very much, Professor Gorman. We do have a hard stop at 11:55 because Professor Gorman has another commitment. So we have 5 minutes for questions. As I mentioned in the beginning, I encourage you to use your raise your hand function because you can speak. It seems that we do have a question from Wei Ming Yuan. I will.
Yes.
Okay.
Can you hear me?
Yes.
I can.
Hi, Professor.
Hello.
Yeah, my name is Yuan. I'm an associate professor in the microbiology immunology department here in CAC. I have a quick question. I saw you showed two plots. One is for the CD19 positive B cells, and one is the total B cell B lymphocytes. And apparently, the total B lymphocytes no clear difference, whereas the CD19 B lymphocytes can see a clear decrease. So, can you tell me a little more about the marker used for the total B lymphocytes?
It's a very good question. The most commonly used markers are CD20 and CD19. If I did not put the marker in the slide is because in the manuscripts, they did not identify the specific markers used to identify B cells. I think the difference between whether you use CD19 or CD20, I don't think is material. I do think that there are differences of the populations that are investigated, where they are in the course of their disease and when they measured the peripheral blood lymphocytes in the course of the disease. And so one of the consistent findings in those studies that did present the B cell data, the differences between the mild and moderate B cell levels versus the severe or critical disease patients, the differences between B cells in mild to moderate versus severe was less than the differences in the total T cells, CD4 or CD8. And this, again, was not consistent in every study. Some studies, in fact, showed no difference in the B cell levels between severe and moderate. Some of the studies showed that NK cells were significantly different, and some of the studies did not. And I think it may be a combination of where the patients are in their disease, as well as the numbers of patients that were studied. All of the studies did, however, show lower levels in severe versus moderate or mild disease, but in some of the studies, the differences were not significant.
Okay, thank you. Thank you. I hope to discuss with you more later when you get more time.
Sure, be happy to.
So,
thank you.
we are running out of time, but we do have 3 questions on the Q&A box. I'm going to try to go through at least 1 or 2 of them. The first question is from Abdelhamed Ramadan. The question is, so calcineurin inhibitors is harmful in spite some studies recommended in treatment of COVID?
It's a very good question, highly controversial. Calcineurin inhibitors, cyclosporin or FK506, have been tried. There were early studies showing that were very compelling and showing outcomes in patients that were on calcineurin inhibitors. In the one of the early studies, unfortunately, I cannot quote the specific investigation or the manuscript. However, recall the study was in recent liver transplant patients versus those with long-term follow-ups following liver transplant. The difference between those patients early on in the course of their liver transplant were on much higher levels of calcineurin inhibitors versus those patients who were later on had lower levels of calcineurin inhibitors. Interestingly, the patients that had lower levels of calcineurin inhibitors seemed to have a better outcome compared to patients on no treatment and no immune suppression, compared to those patients that were on higher levels of immune suppression, where they did not do as well as the patients that were on moderate levels of immune suppression. We also know that steroid therapy is essentially being used in almost all patients being admitted. So I think some level of immune suppression is probably helpful. Too much immunosuppression is probably not helpful. And again, it remains to be seen, we are trying to control this hyperinflammatory response. We also know that cytokine inhibitors, tocilizumab or anti-IL-6 or IL-6 antagonists seem to have some beneficial effect. But again, I think to some extent, it depends when we can catch these patients in the course of their disease.
Thank you. Unfortunately, we're out of time. Professor Gorman kindly offered to answer any remaining questions via email. So if you have access to the USC directory, please send him an email and he will try to answer the question for you. I would like to thank Professor Gorman for the exciting presentation. And we have a few upcoming events. We hope to see you there. Thanks everyone for joining us today, and see you soon. Goodbye.